Hello everyone, thank you so much for joining in. I am your host for the day. I am Deeksha Bhartiya, Director at Genomics, and I would like to wish you all a very happy Teachers' Day. I'd like to take this opportunity to give you a brief introduction of our company Genomics. Genomics is a startup where we are aiming at providing cutting edge research solutions. We provide data analysis and management support, consultancy, and training solutions. So the next question comes who we are. We are research enthusiasts. We are postgraduates, PhDs, and postdocs in our team. We have years of experience in various premier national and international laboratories. Here you can find links to our website and you can connect with us on LinkedIn. You can also connect with us on Instagram, Facebook, Twitter, and also join our WhatsApp group. As I have mentioned in my previous slide, our vision is to provide or to deliver solutions for all types of biological datasets by providing breakthrough inputs. The key competencies of our company are to provide data analysis support, where we have experts in providing bioinformatics support. We also have a lot of expertise in next generation sequencing data analysis. We also have experts for structural bioinformatics. Since we have a lot of project molecular biologists associated with us, we also provide a lot of project consultations. We are also providing solutions for workshops and webinars. We are also providing training programs, short and long term courses, and also we assist in doing dissertation and thesis projects. So without wasting any more of your time, I would like to welcome our speaker for the day, and actually this gives me immense pleasure and it's my honor to invite and welcome the speaker of the day, Dr. Vinod Scaria. He was my PhD supervisor and my mentor. So Dr. Scaria is a clinician and a computational biologist. He is senior scientist at CSIR New Delhi. His laboratory is interested in understanding the function, organization, and regulation of vertebrate genome and how genomic variations could potentially impact them. He is also involved in creating novel methods and resources for analysis and annotation of genomes and understanding the functional impact of genomic variations. He has been part of pioneering collaborative genomics projects aimed at understanding the South Asian and Middle Eastern genome diversity. He has been part of the whole genome sequencing and analysis projects, including the Indian, Sri Lankan, and Malaysian genome projects, and is a member of Hugo Pan-Asian Population Genomics Initiative Task Force. He is also the co-founder of the Genomics for Understanding Rare Disease India Alliance Network, which is also known as CARGEN, which is the pioneer and one of the largest networks of clinicians and researchers in India working on rare genetic diseases. So welcome, sir. First of all, we have a very happy Teachers' Day to you, and I thank you for accepting our invitation. You may now share your slides and we can start your webinar. Thank you all. Stay.
Good afternoon, everybody. It's a pleasure to connect to you online. For the next 20 minutes or so, I will talk to you about the genomics of rare diseases in India regarding experience. And this is where I come from, the CSI Institute of Genomics Integrated Biology. Before I start my talk, let me give a few disclosures and conflict of interest statements, especially from companies for which we work for and in terms of consultancy service. Well, I'll give an overview of the talk, and before I talk, let me first go back to around 2006 to 2008, and we're just fortunate to be starting our lab at that point in time. And that's also one of the most interesting times in human genomics, where, as you see here, the cost of human genome sequencing was dwindling rapidly. But if you ask this question, does this dwindling cost really change the world of genomics? The answer is probably yes, but and also no. And as you see here, you would see there's a large gap in the genomic knowledge in one of the huge admixed populations spanning Middle East and Central Southeast Asia. At that point of time, the 6 genomes that were done, which includes the reference genome, the Craig Benson and the Watson genomes, and the Chinese, Japanese, and the Korean genomes, were all from quite homogeneous or less admixed populations. And what you see here in the map is essentially a gap which encompasses close to a third of the world population. And that's when we started in 2008 to start to sequence human genomes in India. And we put together one of the first human genomes at the end of 2009, making it the 6th country to be able to do so. And the idea of doing this was to be able to learn the tricks and the traits of how to put together a genome sequence and also to make sense out of it in terms of what is clinically useful and what is clinically impactful. We went ahead and sequenced a large number of personal genomes in this part of the world, which includes the first Sri Lankan, the first Malaysian, and we collaborated very extensively with the Pan-Asian Population Genomics Initiative, one of the flagship programs of the Human Genome Program. And very recently, we ventured in the Middle East, trying to create one of the largest databases of Middle Eastern populations called Almina, which encompasses 2,000 whole genomes and exomes. Very recently, we also completed the first 1,000 genomes as part of the Indigen program, sequencing the length and breadth of the country. If you ask this question, where do you really use this in genomic information in real-life clinical applications, I would come back to monsters and sinners, and I'm sure you would have all learned fables or read fables, and you have all heard about monsters and sinners, but in many ways we don't know or we have not even seen many of them. But in fact, today we know many of them could actually be genetic mutations. The monster of Ravenna depicted here, where it looks very similar to what is today known as Robert syndrome. If you put a number to this problem, there are around 300 million people worldwide suffering from one of the other rare genetic condition or a rare genetic trait, and 70 million of them have their home in India. And this is a big problem because a large number of them go through multiple clinician visits, multiple rounds of misdiagnosis, and multiple years, an estimate of around 7 years before even they have a provisional clinical diagnosis. And that's largely because there's a lack of general awareness, there's a lack of easily and affordable and accessible diagnostics, and the lack of understanding of the mutational spectrum, which can make diagnostics very cheap and affordable. But of course, the other way to look at it is to actually look at it in the perspective of the population. And India is a very large country, encompassing almost a sixth of the world population that we know today. Its genetic pool is diverse, unlike other large populations, which are quite homogeneous. It encompasses representative populations from across the world and stratified into small endogamous groups. That necessarily means that the genetic diseases would run with very high frequency in such endogamous groups. And even more importantly, that we have not been displaced by war or famine for the last many centuries, and that still means that we could trace the origins or ancestry of individuals 3, 4, or in some cases even 10 generations. So what we put together was a large collaborative network, which we today call as Guardian. Guardian stands for Genomics and Understanding Rare Diseases India Lens Network. The program has a very well laid out principle. We work with clinicians, specialists who see patients with rare conditions in the clinic. They refer to a standardized process of sample collection, consenting, analysis, and of course reporting. And on an average year, we could report approximately around 1,000 patients with a diagnostic positivity of around 60 to 70%, depending upon the case scenario. That would still mean that we are not reporting to a large number of individuals, and that's what we take up for in-depth analysis and in-depth research through in vitro, in vivo, as well as in silico approaches. And the ultimate aim is to be able to give back a report to every single patient. And today, this is one of the largest networks in the country, encompassing around 280 clinical clinicians from around 70 centers, making one of the largest of its kind in this part of the world. And of course, the outcomes of this could be a large consortium involving a large number of individuals. But the basis of all that I talked is a large clinical automation system which collects clinical information, genomic information, puts together clinical information, provides a priority gene list which can be looked upon by clinicians and geneticists to arrive at a more possible diagnosis. And the backend to this encompasses very large datasets, which includes a large number of supercentenarians who are over 100 years old, 100 years of age, and apparently normal. This also includes family controls running a few thousands, and of course, very recently, the population genome program data in terms of myelin frequencies coming from the Indigen consortium. I'll give a few examples of how this happens. And this is the first example, and this is a family, as you see here, 2 children apparently affected with an extremely rare genetic condition and born to apparently normal individuals. Now, there are two things that you need to keep in mind. And the first is that the children are abnormal and that two children, which necessarily means that the genetic counseling was probably inappropriate. In such cases, what we typically do is to do an exome sequencing. In this particular case, we did an exome sequencing. We did find the mutation in the TGM1 gene, confirming the diagnosis of this condition called lamellitis. We went back and did Sanger sequencing to establish that these variants were real and could be validated. And like for every other family, what the family would require is to have a normal child, and prenatal testing and counseling was offered, and a normal child was born to the family. And to make things even better, these two children with TGM1 mutations were put on retinoic acid, and they improved quite drastically. And this is a photograph that you see 3 months after they were put on retinoic acid. This is another family. This family lost multiple different children in multiple different generations due to sepsis and pneumonia very early in age, typically before around 6 to 1 year of age. And instantly this caught our attention because two children were admitted at the same point in time to the Government Medical College in Kozhikode, which has been a quite long collaborator to this program. And on close workup, what we found is the B cells were abnormally low, sort of confirming this diagnosis, at least in the clinical sense, as a gamma globulinemia, and a type of gamma globulinemia which is typically affected in male children, the X-linked type. We did a genome sequencing for these individuals. What we found was a large deletion encompassing the gene exons of the BTK. Here what we confirm the diagnosis of X-linked gamma globulinemia due to genetic mutations in the BTK gene. And of course, since, as you've seen, the family is large, we could develop a cost-effective diagnostic. Since this was a chromosomal deletion, you could look at MLPA, and this cost-effective diagnostic was actually built and validated and was applied to the entire family. Now, today we have 16 individuals who were tested to be carriers in this particular family, for which genetic counseling was offered. One child, of course, underwent metabolic stem cell transplant, and another child is underway. Of course, we lost one child to the disease because we could not arrive at diagnosis at an earlier point in time. But this gives you an advantage that an entire community and an entire family could avail genetic screening and genetic diagnosis at a quite affordable cost. The family adopted to carry screening and counseling, and therefore provides a unique opportunity to prevent an early and offer early diagnosis for children in this. And today this program in the Government Medical College Calicut has built into a large consortium which today encompasses close to 350 case referrals over 3 years. We have built cost-effective diagnostics which are now implemented in these centers, and the centers offer a holistic approach including case diagnosis, genetic counseling, genetic testing services, and of course community interventions by going back to the community, educating the community, and offering them carrier counseling and genetic testing. This is another family which came to us with a peculiar genetic diagnosis or peculiar clinical diagnosis of periodic liver syndromes. It's a young individual from a quite consanguineous family, as you see here, referred to us from Epigenetic Medical College in Vellore. And this individual didn't have a diagnosis for the last 5 years. The only symptom that he had was recurrent fever. Now, typically, after you do exome and genome sequencing, you sort of get to the diagnosis. In this particular case, the mutations were found in the MLPA gene, confirming the diagnosis of familial Mediterranean fever. And the next generation sequencing also offers you an approach in this particular case to look at compound heterozygous mutations, which would now be validated on Sanger sequencing in the entire family. And as you see here, one of the mutation came from paternal lineage, and the other mutation came from the maternal lineage. And in cross-consanguineous communities, this becomes a frequent occurrence where you could have otherwise extremely genetic mutations which would segregate in families. And since these mutations were extremely rare, we went back and looked at the ancestry of this individual genomic data provides a unique opportunity to go back and look at the ancestry of these individuals. And what we show is that this individual was part admixed in terms of having a mixed lineage, part Persian and part Indian, and therefore making the diagnosis even more confirmed that this is familial Mediterranean fever, because familial Mediterranean fever is a disease which is very prevalent in the Mediterranean basin. And then points to a large number of individuals who trace an ancestry to the Mediterranean basin. Today we have around at least 40 major communities in India encompassing close to 1 million population, and that also highlights the requirement to collaborate very extensively with other countries. In this particular case, we collaborated with a large number of labs across the Middle East to create one of the largest and the most comprehensive datasets for Middle Eastern population genomes, encompassing around 26 million variants, which is published way back in 2007. One thing that we need to keep in mind is that in all of clinical medicine, the basic assumption is that the patient will come to the clinician with an abnormality. The clinician will make a diagnosis, offer the right genetic testing, counseling services, as well as the treatment. But it should always be kept in mind that otherwise traits or phenotypes which are not really detrimental to the individual in terms of his day-to-day functionality could never come to a clinician. There is one example which was referred to as from the SMS Medical College in Jaipur, Professor Sandeep Mathur has been a collaborator. We've tried to follow up this patient for many years now, at least 3 years, and it was almost impossible to reach out to the community and the individuals who are affected because the only symptom that they would have is essentially hypogonadism. And the stigma attached to many of the genetic disorders would prevent patients coming to you. That would mean that you need to have newer approaches to looking at the frequencies of genetic variations and genetic diseases in the population, and the only way you could approach that is to be able to do whole genome sequencing of populations. And that's when 6 months ago we embarked on this program called Indigene. The idea of Indigene was to essentially collect samples which are representative of large populations across the country to be able to offer whole genome sequencing to these individuals with the idea of understanding what is the genetic spectrum of rare genetic variants which are very prevalent in a population, because a number of 1,000 individuals would only give the prevalence of 1 in a few million individuals. The other idea was to, of course, be able to use the genomic data in a population perspective to look at pharmacogenomic landscape, and also use this information to build new and cost-effective and affordable, of course, technologies for commercial applications. I'll give a few glances into what we could do with this data. We looked at the epidemiology of treatable intellectual disabilities, treatable because it offers a new opportunity to treat these conditions if they were diagnosed very early on. Through a very systematic analysis, including variant annotations as per the recent ACMG guidelines, what we figured out was there are around 13 variants which are pathogenic and another 10 which were likely pathogenic, which could be figured out from this large data set of population genomes. We could, of course, look at the allele frequencies of these variants in the population, and of course compare it and contrast it with the global populations. And what this would give us essentially the prevalent genetic mutations which are essentially in a carrier status, typically autosomal recessive conditions where the individuals are carriers for these genetic mutations, and this would provide an insight into what the prevalent genetic variations that could occur in populations, and of course the frequency in endogamous populations, because you know the average pattern. And what we find out is that approximately 1 in 25 individuals, or approximately 4% of individuals in the cohort, were carriers of one or other treatable intellectual disabilities. Now, the advantage of understanding these variants is that now you could offer very cost-effective genetic diagnosis, and the prevalence would also allow us to roll out newborn screening programs to be able to prevent these diseases, not just prevent the diseases by early carrier screening, but also to be able to treat the diseases early on by early diagnosis in the early neonatal period. And from case diagnosis and working with clinicians, we have come a whole circle. There are multiple different programs which add on to the Scaria program of mentioned as the COMED. which offers cost-effective genetic diagnosis free of cost to thousands of individuals who suffer from genetic diseases. At this moment, we have around 130 genetic tests, which have now been tried in approximately around 20,000 tests. Now, this information could now be passed on to the diagnostic industry, where they could learn from the know-how and the experience to be able to build their own versions of cost-effective diagnostics. And today we have worked with multiple diagnostic companies in the country to be able to make genetic diagnostics very affordable as well as evidence-based. And what adds to this impetus is essentially population scale genomes that came from the Indigen consortium, as I mentioned about it. And coming in a whole circle, what's really more important is to be able to now invest into research and drug discovery for rare genetic diseases. And we have extensively been using multiple models, including zebrafish as a model system, as well as high-end computational approaches, including machine learning and artificial intelligence, to be able to arrive at better drug candidates that can be now trialed out in the preclinical and the clinical situations hopefully in the future. I think I'll stop there and take questions, but before that, let me acknowledge a lot of people that make this possible. A lot of students in the lab and students have moved out. Fantastic collaborations of the last decade with Dr. Sridhar. A large number of clinical collaborators without whom this program would not have meant anything. A large number of collaborators from across the world. Vajira from University of Colombo for Sri Lankan genome, and Professor Mohammad Saki Saleh from the UITM Malaysia for the Malaysian Genome Program. And of course, consistent funding to do all this from the Council for Scientific and Industrial Research India through multiple different programs spanning the last 1 decade. Thank you.
Thank you so much, sir. Thank you for such an informative session. I know there were some participants who were facing some technical problems and were not able to hear it properly. Don't worry, participants. Even if you have missed something, we'll share you the recording. Now I'll take this opportunity to share your questions with sir. So now I'm going to there are only few participants who have asked relevant questions, questions relevant to the session. So the first question was that what is MLPA diagnostic assay? Somebody wants to ask that.
MLPA is multiplex ligation probe amplification. So essentially it is a probe which is designed to a specific region in the genome, right? And if it hybridizes, then you can extend it. And then therefore you can read the probe using a fluorescent agent. Now, this is particularly used in copy number changes, where you can look at copy neutral. So for example, if you have two alleles, then you would have a signal. If you have half the allele, that means that one of the allele is sort of deleted, then you have half the signal. And if there is a homozygous deletion, then you have no signal. So in other words, you can look at the copy number changes at a particular locus. So this is locus specific. In a particular region in the genome, you can look at the copy number changes. Now you can multiplex it for multiple probes across a particular genomic region. For example, if you look at a gene, you would probably design 3 or 4, if not more, probes around that region, and you can multiplex it together so that you can do the readout at one run, right? And this is typically run on a Sanger sequencer. We don't sequence, but run it on a chromatogram to look at the different lengths of the probes. Each probe gets a fluorescent signal, and since we already know what is the length of the probe that we sort of put in for the testing in the first place, you know what region in the genome had a deletion or a duplication at that particular length.
So thank you, sir. So now I'll take you through the next question. So next question says, when you say you can treat the detected disease in a population, do you mean you can fully cure the patient? Is it possible to reduce the effects of a genetic disease?
Yeah, so most genetic diseases unfortunately are not treatable as of now. A significantly small percentage of genetic diseases, and to that number around 20 to 25% of genetic diseases today, have either a definitive therapy, like for example Wilson's disease, or a supportive therapy which can ameliorate the condition.
So supportive therapy, for example, could be blood transfusion as in the case of thalassemia or sickle cell anemia. And a definitive therapy, for example, could be enzyme replacement in lysosomal storage disorders, or for example, depenicillamine in Wilson's disease. So around a fourth of the genetic disorders or a fifth of the genetic disorders would have either a definitive or supportive therapy available. But still, majority 75% of the genetic diseases would not have a definitive treatment available.
Okay, so the next question is, what can be the worst case scenario of rare genetic disease? No.
I didn't really get what's the worst case scenario. So I would rather put it in a positive way in the sense of what's the opportunity of a rare genetic disease. And the opportunity of understanding a rare genetic disease is that if you identify the genetic mutation, you can prevent another child from being born with the same mutation. So that is essentially an opportunity.
And can we detect rare genetic diseases before birth or during early pregnancy?
You can identify carriers who are parents who are apparently no genetic disease. You can screen the fetus very early in pregnancy, what we call as prenatal genetic screening. If the couple is opting for in vitro fertilization, we can do something which is called as pre-implantation genetic diagnosis. We can do screening at birth, which is called newborn screening, and we can do screening at any point in life of the individual. So the opportunity of genetic testing is right from before you were born, the time that you're born, time that you're in the womb, and henceforth in your life.
And can rare genetic disorders be treated with bone marrow therapy?
Yes, some genetic disorders can be treated using bone marrow therapy. And one of the conditions that we have been working on quite extensively is a condition called primary immunodeficiency. This necessarily means that the body doesn't have a proper fully functional immune system. And one of the curative therapies for this kind of condition is to do hematopoietic bone marrow transplant. And there you essentially replace your bone marrow with a donor bone marrow, which will populate and produce those immune cells that you want. So yes, for some conditions, bone marrow therapy is the definitive treatment.
Okay. So the next question is, how can be severe genetic disorders be reversed or have minimal effect on individual during the fetal or embryonic stage?
Yeah, so like I said, a lot of genetic disorders could be, at least a fourth of genetic disorders today could be either treated using a definitive therapy or could be treated using a supportive therapy. Now, these can be screened very early on to be able to understand whether the child is going to develop that genetic condition. If the child is going to develop that genetic condition, you can initiate the treatment very early on if they're treatable. And that's an opportunity because then the child could have a perfectly normal life. Like now we have a lot of children with primary immunodeficiency disorders who live a perfectly normal life after bone marrow transplant because they could be diagnosed very early on, either at the fetus stage or early at birth, and they could be administered bone marrow transplant very early on because they would not have otherwise survived without a functional immune system.
Okay. And then there's a question that can there be genomic variations for susceptibility to cancer when these populations come in contact with certain toxic chemicals or metals?
Yeah. So yes, absolutely relevant question. Not all cancers are caused by germline susceptibility. Some cancers are caused by germline susceptibility. The best examples would be BRCA1, BRCA2, or breast and ovarian cancer, mammalian breast and ovarian cancer. Now, having BRCA1, BRCA2 genetic variants which are pathogenic doesn't mean that you will develop cancer, but if you have a familial history of cancer and you have BRCA1, BRCA2, and you're also exposed to the right kind of environmental insults, then your chance of developing cancer is very large. So there are genetic predispositions for cancer, quite a lot of them. The incidental gene list, the 59 odd incidental gene list, which we provide to these individuals who have done sequencing, essentially covers these incidental findings, which otherwise you could be a perfectly normal individual. You could have a genetic variant. If rightly exposed and if rightly predisposed, then you might have a higher than normal chance of developing cancer.
So now then, next question is that is the Guardian project covering epigenetics as well?
So at this moment, the Guardian program doesn't cover epigenetics. There are epigenetic diseases, very few at this moment. So at this moment, we don't cover.
Okay, and then there's a question by, uh, in fact, one of our trainees who's asking that how willing are endogamous populations to share, how willing are these endogamous populations to share their genetic data, and does it pose any risk in future to the community that their genetic data is revealed? Can this technique be used to identify a population which has potential to become non-progressor of HIV? So
Okay. Yeah, so very interesting question. So endogamous communities essentially mean that you marry within the same community,
exactly.
right?
Yeah.
And frankly speaking, practically all communities that you sort of know about marry within their own community. You can just look around yourself and including your own families, and you will realize that a significant large majority of the population are from endogamous communities and they marry within the same endogamous communities. Now, there are two problems to this. One problem is that if the community is very small, right, and then you have a genetic disease which is very predominantly present in this community, it might actually result in discrimination. Discrimination when you marry from a different community. But by definition, endogamous communities mean that you marry within the same community. So probably that is not going to be a major problem here. But in communities which are small but are outbred, it might actually, or are now becoming outbred, yes, it might cause a problem in discrimination. And then therefore we typically would not want to reveal the community or the community's name in such conditions. What was the second question?
So the second question was, can this technique be used to identify a population which has potential to become non-progressor of HIV?
So, so there are genetic conditions or genetic mutations, essentially CCR5 delta, which are genetic variations which protect individuals from developing HIV. Now, the allele frequencies for this particular genetic mutations are variable across populations, but there is no population where this is a major allele which can sort of identify the population as resistant to HIV. I think the highest allele frequency in any Indian population is 1 in 100, if I'm not wrong. So that is so small that you would not want to say that this community is resistant, though there have been news reports on that particular community. By numbers, it's actually a very small number who are actually resistant.
Okay, so the next question is then that does epigenetic factors have any effect in impacting these rare genetic diseases?
Absolutely. So one best example is sickle cell anemia. So sickle cell anemia is caused by the HBS mutation, one single genetic mutation, and this is the same genetic mutation across the world. Now, all children who have sickle cell anemia mutations don't have the exact severity of genetic diseases, right? Some sickle cell patients develop sickle cell crisis, which is a much severe condition. Some children who have the same genetic mutation homozygous state might be apparently normal, might not require transfusion, and might live a normal life. And most of this is essentially because of two factors. One is what we call as modifier locus. Modifier locus necessarily means that the additional genetic variations, either in the vicinity of this first genetic mutation or far away from this first genetic mutation, which can now influence the penetrance or the phenotypic nature of this particular mutation. Now, one way that this additional genetic mutations could influence a phenotypic outcome is through epigenetic modifications.
Okay, so now we have a lot of questions now. So in the interest of time, I'll take and another 2-3 questions only. So now I'll have this question that how are SCIDS, SCIDS patients at adult stage treated? Only by bone marrow transplant or by injecting cultured cells in the tissue or by at this by enzyme replacement therapies?
So, if you are talking about SCID as severe combined immunodeficiency,
Yeah.
most of the children with severe combined immunodeficiency will not, if not die early on, they typically will die early in their childhood.
Okay. So
because they don't have an immune system, and they're predisposed to practically all infections that are around.
okay. So the next question that I'll take is, once the variant or rare genetic disease is identified, will gene therapy be useful at that time? That is at any period, time of life period. So
Very interesting question, because gene therapy is something that is hot nowadays. It has been hot before, it is now hot again, thanks to President Gas. Yes, so the answer is yes and no. It depends upon what tissue does the genetic condition affects. Now, if the genetic condition affects a tissue, like for example the brain, early in development, then very unlikely that you can change it using gene therapy. Of course, there are some exceptions to that, but largely terminal tissues which are differentiated, very unlikely that you can treat using gene therapy. But now, if you, for example, think of a regenerative tissue, the examples of regenerative tissue would be, for example, liver, you could say bone marrow, you could say skin. Yes, since the skin can regenerate, you can have engineered skin, which can now replace the abnormal skin over time. And this has been tried for conditions like, for example, epidermolysis bullosa. This has been tried for example, blood disorders, where the bone marrow can, a normal cell can actually repopulate the bone marrow, and then therefore you can treat such conditions. So it depends upon what is the tissue that the genetic disease affects, and if the tissue is a regeneratable tissue, then in most cases, yes, you could possibly treat them using gene therapy. But having said that, there are very few gene therapies which are currently available. In the future, we will have far more of them.
okay, so now I'll be taking last. So I'll take the last question for the day now, so that we can just take in one last question, which was interesting. That what are the current loopholes regarding genetic diseases in India, and what could be the reason?
The biggest problem with genetic diseases today in India is that most patients do not have a genetic diagnosis, right? And most people do not have a genetic diagnosis because the awareness that a significant number of genetic conditions are treatable if diagnosed appropriately is not there. That is point number one. Point number two is, even if they have a genetic condition and a clinician sort of makes a diagnosis of the genetic condition, accessing the molecular diagnosis to confirm that is not readily available. Yes, there is labs which are offering genetic testing, but it is still much beyond the affordability of a normal individual, according to the what you call the GDP that we have. And third is, of course, assume that the individual got the diagnosis, got the genetic test. Getting the therapies in a sort of affordable and accessible way is still a big problem in India, because many of the therapies are quite expensive that most patients cannot afford. Yeah, so it is actually problems everywhere. So it is not easy to be a patient with a genetic condition.
Okay, so I think these were the last few questions, and thank you so much, sir. Thank you so much for attending. So there are a few participants who are facing some problems in filling in the feedback form. Don't worry, participants. The feedback form will be still be working for another 20-25 minutes. You'll still have some time to fill in that one. So and we'll deal with the technical issues later on. So before that, let's say thank you so much to sir, and we should thank him for giving such an informative session, and thank you so much, and we'll love to hear from you again.
Thank you for organizing this talk, and I'm sure there are probably more people with very pressing questions. So this video, I believe, would be available on YouTube, and you're welcome to actually come back and put your comments, and if possible, I would be able to address them on YouTube.